There's a lot of possibilities. In the past, we have used for expansion IL-2 only, and we know that with a cytokine cocktail, IL-2, 50, 80, 21, you can really increase the expansion rate 10, 100, 1,000-fold more compared to the past. You can make primary human NK cell, as an example, on the automated clinical protege, what we are doing together in a nice cooperation with Milteney Biotech, and there are protocols for purification and then transduction and expansion of the NK cells. But you can also do it manual in live bags and others. There are a lot of opportunities, and you can clearly use them as off-the-shelf product. You can also make them iPS-derived NK cells for off-the-shelf product. There are a lot of opportunities in the manufacturing protocol.